All right, this video is going to be about the second part of the oxidation of glucose, which is the citric acid cycle. So we just talked about glycolysis. Glycolysis occurred in the cytoplasm. It was anaerobic, so it didn't use oxygen. But it did provide us with a molecule called pyruvate. Two of them. And this was a 3-carbon molecule. And if oxygen is available, pyruvate can go into the mitochondria. So this is going to happen in the mitochondria, specifically in the matrix. So if you remember, the mitochondria, it's this little organelle, looks kind of like a bacteria, and inside of it, it has a second membrane and had all this like cristae in this membrane. It's all kind of folded or whatever. The center of this mitochondria is what we call the matrix. So this is where the citric acid cycle is going to happen. So before we can enter the citric acid cycle, we have to change the pyruvate because the citric acid cycle requires a molecule that's only 2 carbons, and this one is 3 carbons big. So we have this little transition reaction that occurs. Transition reaction occurs when the pyruvate comes into the mitochondria. And what happens with the pyruvate is it goes through some reactions and it decarboxylates. That's one of the things that happens to it. And from that, we lose our carbon as carbon dioxide. So we get rid of the CO2. Another thing we get out of this reaction is an NADH. So we get an NADH from this reaction. Once it's over, we add within this thing a coenzyme A. And so what we end up with is a 2-carbon molecule with a coenzyme A called acetyl-CoA. And this acetyl-CoA can go into the citric acid cycle. So once that happens, it goes in, and what it does is it immediately binds with a 4-carbon molecule called oxaloacetate. Oxaloacetate, or OAA. And when this one combines with acetyl-CoA, we end up with something called citrate. And citrate is a 6-carbon molecule. 1, 2, 3, 4, 5, 6. And you can see that this citric acid cycle, which is what this thing is, citric acid cycle, citric acid cycle, and it goes around like a wheel. And so we're starting at citrate here, where we combine the oxaloacetate and the acetyl-CoA, and it's going to go around, and we're going to break a couple of these bonds here from this 6-carbon citrate and get back to oxaloacetate. So this citrate, in this first sort of reaction, it has to be sort of changed around so that it can be cleaved so that the carbon can be worked on. So from citrate, we get isocitrate. And not much happens here. We still have a 6-carbon molecule. 1, 2, 3, 4, 5, 6. Okay. So we still have a 6-carbon molecule, but from isocitrate to the next intermediate, which is called alpha ketoglutarate, alpha ketoglutarate is a 5-carbon molecule. 1, 2, 3, 4, 5. So to get from isocitrate to alpha ketoglutarate, and all of these are reactions, and they're being caused by specific enzymes, which we're not going to worry about that right now. So in this reaction from isocitrate to alpha ketoglutarate, we're going to have a decarboxylation. So we're going to lose a carbon dioxide. So we're going to lose this carbon. And in this reaction, we're also going to, the right color here, we're going to get an NADH. NAD to NADH. Okay. From alpha ketoglutarate to the next intermediate, the same thing happens. We have this 5-carbon molecule, this alpha ketoglutarate, and the same thing is going to occur, and we're going to end up with succinyl-CoA. Succinyl-CoA. And to do that, we're going to go through a decarboxylation, get a CO2, get rid of this carbon. So now we have a 4-carbon molecule. With this as well, also going to get an NADH. Okay, so from succinyl-CoA, we're going to go on and we're going to get succinate. And in that reaction, we're going to end up with, or getting, an ATP. So we're going to end up going through substrate level phosphorylation and converting an ADP, adding a phosphate, and getting ATP there. So substrate level phosphorylation. And remember, that's just direct taking a phosphate and sticking it on an ADP, making it ATP. So now we have an ATP. So from succinate through the next reaction, we end up with something called fumarate. And in that reaction, we end up with an FADH2. So FAD to FADH2, which, if you remember, was another one of those electron carriers like NADH. So we've got one of those. The next reaction changes the molecule. And remember, we're still 4 carbons here. All of these. Okay, so the next intermediate is called malate, and we're still 4 carbon. And from malate back to oxaloacetate, we get another NADH. And we're back to oxaloacetate. So that's the process of the citric acid cycle. And if you look at it closely, what are we getting out of this thing? We're getting quite a bit. Let me erase my little mitochondria really quickly here. So I can have some space. All right, so what are we getting here? So we'll put like the totals here. Okay, well, don't forget about this transition reaction. So the transition reaction that occurred here. We had a pyruvate, we got it to acetyl-CoA, and with it we got an NADH. And we got two of them because remember, we have two pyruvate. That's how many we made in glycolysis. So through the transition reaction, we get two NADHs. Okay, and then if you count up through the citric acid cycle, we have 1, 2, 3 NADHs, 1 FADH2, and 1 ATP. And guess what? There's 2 acetyl-CoAs, so it's really 6, 2, and 2. So the grand total of stuff that we get out of the citric acid cycle once pyruvate goes into the mitochondria is 8 NADHs, 2 FADH2, and 2 ATP. So that's the grand total of stuff we're going to get from the citric acid cycle. That doesn't seem like a lot. We got 2 ATP. Ooh, big deal. Well, guess what? All these electron carriers that we were talking about before, what are we going to do with those things? Well, we can take these electron carriers that we've accumulated, and remember, we got 2 of them from glycolysis. Remember, glycolysis, we got 2 NADH and 2 ATP. So now we have 4 ATP, and we actually have 10 NADHs, 2 FADH2s. And we can take those electron carriers over to a place within the mitochondria, and we can convert those high energy electrons into ATP. And that's what we're going to talk about next.